Our next presentation is MeiraGTx.
Hi everyone, I'll let people come back from lunch. My name is David Rubenstein. I'm head of BD at MeiraGTx. A few forward-looking statements. MeiraGTx is a gene therapy company. We solely do AAV gene therapies. One of very few companies in the field that truly has end-to-end capabilities from early discovery work all the way through to having the ability to manufacture AAVs, both for clinical and commercial. We've built one of the largest clinical pipelines in gene therapy. So currently we have 6 programs in the clinic across different therapeutic areas. Of these 6 programs, 3 of them are in late-stage development. We have 4 programs in ophthalmology. That's where we started from. But we also have a Parkinson's program that successfully completed a phase 2 study, and we're very excited about gearing up towards a phase 3, as well as a phase 2 study in xerostomia and Sjogren's that is currently, again, in phase 2 that might be registrational. I mentioned manufacturing. We brought in all the key elements for AAV manufacturing, so not only viral vector manufacturing, but also plasmid and QC, and that enables us to really speed programs into the clinic. It's worth noting that we are the commercial manufacturer for Johnson & Johnson, and last year Janssen acquired our phase 3 retinitis pigmentosa program, and Meira is the commercial manufacturer for Janssen. We have next generation vector optimization technologies. One thing worth mentioning here is our promoter group. We have one of the largest efforts in the industry to identify novel promoters, and I'll talk about that a little bit. And then I think one of the holy grails in gene therapies has been the ability to control a transgene once the AAV is delivered. So really in the past 8 years or so, MeiraGTx has developed a proprietary system based on Ribo-switches that allows you to switch on and off gene expression in a very precise manner with oral small molecules, not only in a binary way on and off, but also in a very precise dose-responsive manner. I won't go over our pipeline. You can just appreciate that we have lots going on, 6 programs in the clinic, a lot in preclinical stage across multiple therapeutic areas. We're about 370 people, publicly traded company. We're headquartered in New York City, but we have a large presence also in the UK and a large AAV manufacturing facility in Ireland. Last year, since this is the partnering conference, we made 2 notable deals, one with Janssen, where again we sold our phase 3 RPGR program for $415 million. Meira remains the commercial manufacturer for this program. And a second deal that we did last year was with Sanofi. Sanofi invested $30 million in Meira in exchange for a right of first negotiation for certain Ribo-switch targets, as well as our xerostomia program. Again, since this is a partnering conference, there's multiple ways we work with different companies, from developing novel gene therapies for targets of interest, whether it's traditional gene therapies or Ribo-switch regulated, as well as a few pipeline programs that are interesting for collaboration. I mentioned our Parkinson's program that's entering a phase 3 study, our Sjogren's and xerostomia program that's currently in a registrational phase 2. We have a broad pipeline for ophthalmology, both IRDs and non-IRDs, and we're free to partner after our 5-year collaboration with Janssen has ended. And lastly, we have a very interesting program in ALS, a first-in-class program that allows us to treat not only the 10% of patients that are genetically, that have genetic ALS, but also the 90% of patients that have idiopathic ALS. So I'm happy to talk about any of these programs, but today I'll also only focus on Parkinson. Gene regulation is a major focus for the company, and 2 buckets, 2 very powerful levers that we can pull is transcriptional control, which is driven by promoters, and promoters really allow you to control where your gene is expressed in very specific cell types and to what level. And the second lever that I mentioned is temporal control, when your transgene is expressed and to what level, using Ribo-switch. And both of these levers work together to allow really unprecedented control of genetic medicine. Very briefly on promoters, we have a large promoter group that combines rational design of promoters with high throughput approaches. And on top of these approaches, we overlay AI and machine learning to allow in silico mutations of these promoters that we identify in the lab to come up with even better performing promoters. That allowed us to build a very comprehensive library of promoters, from very small promoters, shorter than 200 base pairs, to very cell-specific promoters. And you can see the different tissues, from ocular to CNS, muscle. We have a muscle promoter that is 20 times better than the muscle promoter being used in the clinic. And that's really important because it allows you to down dose the viral dose that you need to achieve the same level of expression. Switching gears to Ribo-switch, the very high level description of the system is that when you deliver your vector, and it could be viral or non-viral, with a regulation cassette, the default state is off, and we can actually achieve complete off state with no expression of transgene whatsoever. And then only in the presence of a small molecule inducer that is taken orally, we get very robust activation of gene expression in a dose-dependent manner. We achieve unprecedented dynamic ranges in the 5,000 to 10,000 fold. So really, this is the first time where a company is able to achieve precise control of gene expression after a gene therapy is delivered. We apply this across many, many different use cases, from vectorized antibodies to cell therapy. In cell therapy, which is one of the amazing use cases of this, we control the expression of CAR, and this really allows us to circumvent some of the disadvantages with cell therapy today, like tonic signaling and exhaustion of T cells, and we show that the same level of tumor clearance could be achieved with a third of the dose of the CAR T. Another exciting area is therapeutic hormones and peptides, where we control, again, the precise levels that are produced by the AAV, and we now combine different metabolic peptides like GLP-1, GIP. We combine these with different cytokines to preserve muscle mass. So this is something that is part of our deal with Sanofi. They have a right of first negotiation there. And then lastly, DNA editing is another interesting aspect of this, where some companies deliver the nuclease with an AAV, which means that the DNA gets edited over and over again, resulting in a potential for off-target editing. So what we do is we transiently express the nuclease, we achieve efficient editing, and then we turn it off, and it goes away. This is a partial list of all the genes that we regulated to date. You can see all the antibodies, the different CAR constructs, and this is just to give you a sense of how modular the system is. So it currently takes us about 3 to 4 months to take any transgene and convert it into a regulatable format. So this is a highly modular system that we apply to, I think, now close to 30 different transgenes in different contexts. I won't go through the data. We have really mountains of data. This is just to show you in vitro data of vectorized antibodies, and you can see some of the more familiar antibodies here, and you can see a very nice dose response to increasing levels of the inducer. And just to mention again that we're working on combination of different metabolic peptides and hormones, like this group of GLP-1s together with other peptides. The nice thing about this is that we can use the natural short-lived version of these peptides instead of trying to engineer them to be long-lived, which has been the approach of the industry. So I wanted to tell you a little bit about our GAD program for Parkinson's program. It's a really unique program. Again, the only gene or cell therapy to complete a sham-controlled phase 2 study. We're now gearing up towards initiating a phase 3 study. Unlike some of the other gene therapies in gene therapy, we don't focus on specific driver mutations like GBA1, but we can potentially address all of Parkinson patients that stop to adequately respond to dopamine therapy. The statistic today says that because Parkinson is a progressive disease, about half of patients stop adequately responding to oral therapy within 5 years of treatment. This is really the target patient population that we're addressing. What we're doing here in this very complicated schematic, what you need to know is that in Parkinson brains, the loss of dopaminergic neurons leads to an overactivated subthalamic nucleus, here in green. And this is due to an imbalance between glutamate and GABA. What our therapy does is locally deliver small doses of AAV2 that express the enzyme GAD. GAD converts glutamate to GABA, and as you can see on the right, in terms of the mode of action, what we seek to do is to normalize the activity of the subthalamic nucleus and restore motor control. As you can see, again, from the schematic, the subthalamic nucleus and the signaling pathway lies downstream of dopamine signaling, so we can really circumvent dopamine and target directly the subthalamic nucleus. So this is a brief procedure. It does require a neurosurgeon to deliver the AAV. This is a routine procedure that is done by neurosurgeons. It takes about 10 minutes of OR time. It doesn't require even general anesthesia. So we conducted a phase 1 study and a phase 2 study. I'll just give you a taste of the phase 2 study results. In terms of safety, this product has really a magnificent safety profile, so there were no SAEs that were related to the drug. There were only mild adverse reactions that were associated with surgery and resolved after time. Interestingly, one of the key safety measures that you measure in Parkinson is this worsening of Parkinson's disease, again, because this is a progressive disease. And control subjects, 35% of them reported worsening of Parkinson, which is something that you'd expect, but only 0% of patients reported worsening in the treated group. In terms of efficacy, the study met the primary endpoints, which were improvements in motor symptoms at 6 months, but also we saw improvements in a large list of secondary outcomes like complications from dopamine therapy. If you look at these results from the phase 2 and focus on the middle graph here, this is responder rates. We chose a very strict responder rates of 9 points or more in UPDRS Part 3. And you can see that the treated group, we had much higher responder rate, both at 6 months and 12 months compared to placebo. On the right, some of the secondary outcome measures that we measured were sort of these side effects due to L-DOPA treatment. So patients get dyskinesia as they use higher and higher doses of L-DOPA. And you can see that throughout the 12 months, there's a nice separation between the control group and the treated group who experienced much less side effects of the therapy. A really unique aspect of this therapy is that this is the first, I think, ever treatment or clinical study in Parkinson that uses an unbiased biomarker that we developed. So we looked at FDG PET imaging of Parkinson patients, and in an unbiased manner, we came up with this pattern that really highly correlates with clinical response. So the way the brain looks like for a patient that received AAV-GAD and responded to therapy looks completely different than that of a patient that received placebo and might, for example, report an improvement in symptoms because of a placebo effect. So this is, again, the first clinical study that incorporates an unbiased biomarker that we developed. And it also allows us to show a rewiring or recircuitry of the brain in response to therapy that is consistent with mode of action of our therapy. So this is all I had today. I'll just say that this Parkinson program is currently, again, completed the phase 2 study. We're now running a small bridging study with 14 patients using higher dose of the AAV. Preclinical data supports the safety and the potential for improved efficacy with this higher dose. And we'll begin talks with regulators around initiation of a pivotal study. So happy to talk about any of these programs. I'll be around, and don't hesitate to ask. Thank you very much.